Hello, and welcome to this webinar by Asuragen in conjunction with LabRoots on next-generation sequencing lessons in translational diagnostics, mining and migrating robust cancer biomarkers to unified DNA and RNA NGS panels. My name is Gary Latham. I'm the Vice President for Research and Technology Development at Asuragen, and I'll be one of your speakers today. I'll be joined by my colleague, Dr. Brian Haynes, who's a Senior Scientist in Computational Biology within Asuragen's Bioinformatics Department. So what we'd like to cover in this webinar today is to share some of the lessons that we've learned over several years of developing, validating, and implementing NGS-based tests for clinical use. And we'd like to do this in 4 parts. Part 1, laying the foundation. How do you wire together a comprehensive and integrated targeted NGS system for clinical applications, for routine patient testing, and also to advance drug development and support clinical trials? Part 2, turning on the light. Biomarker discovery is an intrinsically complex endeavor. It's made more difficult in challenging clinical specimens such as FFPE tumor biopsies. In this part, Brian will describe how we've used whole transcriptome RNAseek methods to identify RNA biomarkers in FFPE. Part 3, connecting the dots. Identifying the biomarker is really just the first step, and a key subsequent activity is how to take genome-scale methodologies and those markers and migrate them to small footprint assays that can be run routinely in the clinical laboratory. In this segment, Brian will describe how we've been able to develop targeted RNAseek assays that incorporate these novel biomarkers, and how we've been able to migrate them from FFP even to a novel sample type, in this case, fine needle aspiration biopsies. And finally, part 4, putting it all together. I'll wrap up by describing how we've taken DNA biomarkers and RNA biomarkers, including novel markers, and incorporated them into a unified total nucleic acid-based clinical NGS workflow, where we can move beyond the value of any single marker and instead query pathway dysfunctions that are relevant for a range of different clinical questions. We'll then have time for conclusions in each of these segments, and then open the floor for questions. If you do have a question, there's a Q&A box at the lower left of your screen. You're welcome to enter questions in at any point during the webinar. We'll certainly try to address as many as we can at the end, and for those that we can address, we'll certainly follow up as necessary. Part 1, laying the foundation. In this segment, we'd like to describe how to build and implement a comprehensive and targeted NGS system for translational diagnostics, and what some of the key considerations are in this process. Clinical testing requires tight integration across all facets of the workflow, yet commercial NGS reagents are often provided for research use. As such, this puts a burden on clinical laboratories to patch together different components into a workflow that can yield less than desirable results. So our mission at Asuragen has been to optimize each and every component of the workflow and then link them together in a way that can meet the clinical requirements for NGS-based testing. For example, for sample stabilization, Asuragen has an FDA-cleared RNA retain reagent that stabilizes RNA in clinical specimens and can help protect that RNA for downstream assays like targeted RNAseek. For DNA, RNA, or even total nucleic isolation, Asuragen scientists have expertise that dates back to their Ambion heritage in developing technologies for biofluids, for FFPE, for fine needle aspiration samples, and those technologies have been significantly advanced over the past few years. In areas where there have been lapses in these workflows, like in-process QC metrics and assays, we've developed those assays where necessary. And one of the ones I'd like to talk about today is QFI DNA assay, as well as a QFI RNA assay. And then target enrichment, our focus has been on PCR to enrich for targets of interest because it's so well vetted for diagnostic purposes. We've built large-scale multiplexes using this methodology that can ensure even coverage. And then ultimately channeling this information into the analysis pipeline to be able to make confident calls that are relevant for clinical applications. But in addition to linking all these pieces together, it's also important to integrate across different segments of the assay. And one of the points that I'd really like to highlight today is how our ability to interrogate the quantitative characteristics of the sample itself, that information can be used to inform the analysis pipeline so we can get much more reliable data. This slide provides a workflow of this process from pre-analytical to analytical to the post-analytical phases and adds specifications on top of what I mentioned previously. For example, in the pre-analytical phase, the QFI DNA assay allows us to really understand how much of the sample we have that's actionable at the DNA or even the RNA level, and how that can be input into the analytical phase of the reaction to generate reliable results. At the analytical phase, we typically use a range of different enrichment technologies based on PCR, but I think the key point is that we've developed these technologies to accommodate the worst possible scenario of sample, so that once we've built these assays, they can be applied to FFPE or even biofluid specimens. And then ultimately the bioinformatics is to consider all of this information, including the noise that is intrinsic within a given clinical sample, to be able to make the right interpretation. Another key feature is in controls. So although we and many in the community find, for example, purified DNA from cancer cell lines to be very helpful, and they certainly are, we do favor using residual clinical materials whenever possible. And for this reason, Asuragen has access to a 100,000 member FFPE tumor bank for assay development and for validation. So let's put a spotlight on the pre-analytical phase. What are some of the technical challenges that face development of NGS assays? And certainly one of those challenges is with the sample itself, and particularly FFPE specimens. So these samples are known to have relatively low DNA and RNA quality. So of course, you look at these different blocks, you can't possibly tell which one has low versus slightly higher quality. Methods like spectrophotometry or nanodrop are equally blind, since they report just bulk nucleic acid and tell you nothing about its functionality in downstream assays. Fluorescence-based assays like qubit are more informative, but actually they're just as blind for the lowest quality samples that pose the greatest risk to misinterpretation in NGS assays. But there is a method that works and works well. It's called the QFI assay. It's one that we've developed specifically to address this gap in sample characterization going into NGS assays. So QFI stands for the Quantitative Functional Index. And what the QFI assay does is it measures the fraction of amplifiable DNA in a sample. So a QFI assay score of 3% will correspond to 3 amplifiable templates in a background of 100 DNA templates. When we compare the QFI assay score to the relative DNA concentration measured using like a qubit assay, you can see there's a general relationship between these two assays for the higher quality DNA samples, but this relationship falls apart for the lowest quality samples. So once you drop below about 3% in the QFI assay score, qubit is no longer informative, even though the QFI assay itself has a very broad dynamic range you can quantify well below this point. The challenge here is that it's these low quality samples that typically are most at risk to generate false negatives and false positives. And I think a clear demonstration of this fact is shown in this slide, which summarizes data that we generated across 44 FFPE tumor specimens that were profiled using the AmpliSeq cancer panel. These samples represented a range of different DNA quality. Each point in this plot represents a distinct sample. And for the samples that have relatively high quality, high QFI assay score, the number of variants, generally between 10 and 15, is consistent with expectation. However, once you drop below about 6% QFI, at that point, there's a sharp rise in the total number of variants that are detected, in this case, using the ion torrent caller. The reason for this is that you're moving into a range where the assay is less robust, and there can be stochastic calls, particularly near the limit of detection, that flare up. So it's critical to understand the nature of the sample because bioinformatics can treat this. This is not an eyesore or a problem in interpretation that can't be remedied. It can, but it can't be remedied with a flat threshold. It takes a different approach. So we've applied the QFI assay to a broad expanse of specimens, and this slide summarizes the assay results from nearly 2,000 residual clinical FFPE samples. 13 distinct cohorts, and the majority of these are drawn from working with pharmaceutical customers. So as a result, there are two conclusions that can be taken from this slide. The first conclusion is that FFPE DNA is generally of low quality. So the median QFI assay score here is less than 10%. The second conclusion is that there's a broad range of quality, and it can vary by as much as 25-fold from cohort to cohort. For example, this cohort represents relatively high quality for an FFPE sample with a QFI assay score of 26%, whereas this cohort represents a much lower score of less than 1%. Challenges for DNA in terms of quality also pertain to RNA. And the summary here shows with FFPE RNA that across 4 different measures of quality or quantification, the greater than 200 nucleotide fraction on the bioanalyzer compared to nanodrop, qubit, and the QFI assay. The QFI assay by far is the best correlation with the unique exonic content that maps. In fact, there's a relationship between not only QFI and the quality, but also the value of increasing the input as a way to recover and rescue samples and generate more useful information, as shown in the plot here at the lower right. This also pertains to DNA. So we know that if we increase the input from DNA for low quality samples, that we can rescue false negatives and also help cure false positives that can occur. So moving on to the analytical phase, there are a number of different questions that are important in the clinical realm in this particular area. What's the workflow you're going to use? What's the content that's appropriate for either routine clinical testing or for clinical trial work? What is the best platform, or can you use multiple platforms together in a way that can be more valuable than any one on its own? What are the right controls to use? And ultimately, how can you build a workflow with the right content on the right platform with the right controls to have a turnaround time that's responsive for both patient testing and for drug development and clinical trials? So one of the panels that I'd like to highlight here is our Chirasika 500 panel that we've developed specifically for clinical purposes. This is one of more than a dozen of such panels that we've developed. It's the most mature. It targets 17 oncogenes and represents more than 500 mutations indexed in COSMIC, which is why it's called Chirasika 500. This is a panel that we've implemented into our clinical laboratory in accordance with the CAP checklist for next generation sequencing, and it's been optimized to achieve high analytical sensitivity and high PPV, down to about 5% variant detection in FFPE samples across a range of different quality, including some of the more problematic, very low quality samples. In terms of input, well, it can be as low as a nanogram, but again, we don't really think about bulk input because the relevant factor to consider is copy number and template complexity. What is the repertoire of actionable templates that will go into the workflow? I'd like to show how we kind of put the pieces together as it pertains to our CLIA validation of this particular panel. And rather than using mixtures of cancer cell line DNA, our approach was to use residual clinical FFPE DNA. But you can't just mix any 2 samples. It's critical that these are matched by the QFI assay score. Otherwise, you'll have a disproportionation of template quantities in the 2. And when you mix them in a certain ratio, you won't get the expected results. In this case, we compared 2 different samples that actually had a matched QFI assay score of 9% so that they could be mixed in an exact mathematical proportion to give a variant abundance. And one of these samples had a PIK3CA and a KRAS mutation. The other sample had a BRAF V600E mutation. And when we mix them at different levels, what we're able to show is that going down near the limit of analytical sensitivity, a theoretical 4.4% variant was detected at 6%. A 4.5% KRAS variant was detected at 4%. And in the converse titration, a BRAF at 4% was detected at 4.8%. So this is the relevant experiment because this is the exact clinical specimen that's being evaluated in the clinical context of how this NGS assay is implemented in our clinical lab. So moving on to the post-analytical phase, we understand that obviously there's a lot of complexity here, but within complexity lies a lot of opportunity for innovation. So going through the various steps of this workflow to demultiplex, to align to the reference genome, assess the coverage, and then define variants that can be detected with confidence and ultimately outputted in a patient report requires that we consider each of these steps with their own incremental QCs and with an understanding of the sample itself. So to give you an example of how we've used different components of this workflow and information outputs to help inform the informatics, consider two samples. The sample that's shown at the top here is a QFA assay score of 43%. This is a high-quality FFPE DNA. Across the 6 possible two-base hypotheses, you can see that there's an elevation in the variant background that's associated with CGTA transitions. If we compare that with a sample that has a 10-fold lower quality, in this case a QFI assay score of 4%, you can see a general trend that's similar among these various hypotheses in terms of their variant background, but now there's an exaggeration, there's an extreme inflammation in the variant backgrounds associated with the CGTA transition. It's much, much higher. And I think in the personalized medicine age, where we treat each patient as an individual based on their individual tumor profile, it's important to remember that behind each patient is a sample. And in the same spirit, it's critical that we treat samples individually. Clearly, it doesn't make sense to apply a flat threshold across variants for each one of these hypotheses. They have to be adapted to the specific characteristics, the quantitative characteristics of the sample. And that's the value of using information like the QFI assay score to build that information into the bioinformatics pipeline. So we've rolled all this information together in a very user-friendly HTML5 interface called Chirasika. This allows the users to browse through all different domains of the data, from the coverage to QCs to the variant calls, etc. And we find this to be a very helpful resource for how complex data can be distilled and interpreted. Importantly, this taps into the fact that we've not just used a hodgepodge of publicly available tools, but these are innovative bioinformatic algorithms have been built based on profiles of the clinical samples they're intended to test, like FFPE. And as a true measure of that integration, this is showing you just one of the outputs from Chirasika. It's not just the conventional coverage information that you'll get, but also the pre-analytical sample QC, like the QFI assay score, is hardwired into this report. This is a sample that's demonstrated with a very low QFI, has about a 3% QFI. So this is exactly the type of sample that would be likely to generate a large number of variants in downstream NGS. However, if you look at the final output from this particular sample, rather than dozens or even hundreds of variants, we've been able to distill down to a single variant, a single cosmic NRAS mutation. You can see it in a pile up here at 14%. So this is the level of clarity that can be observed when one uses pre-analytical information to power the bioinformatics. So I'd like to wrap up at this point with what we've learned. Well, first and foremost, it's garbage in, garbage out. You have to understand the sample. Nanodrop is essentially a random number generator. Cubic is better, but not nearly good enough. And methods like QFI DNA assay and a QFI RNA assay for RNA can be the most useful metric that we've found to date, and has been extensively tested with up to 2,000 samples and with a great deal of NGS outputs to validate it. It's also important to use controls that are truly representative of the specimens you're going to test, and we've really focused on FFP for that purpose. And finally, you should never do bioinformatics in a vacuum. It's really critical to leverage the pre-analytical sample data and other inputs across the workflow and QC metrics to make sure that you can draw the right conclusions. So I'll wrap up at this point and turn it over to Brian for the next segment.
Thank you, Gary. This is part 2, Turning on the Light. In this segment, I'll discuss how to shed light on the dark matter of cancer through the application of RNAseek to FFPE tumor biopsies. It is well appreciated in cancer that the distribution of mutation frequencies follows a power law distribution, sometimes referred to as the long tail of cancer. TCGA and other consortia are powering their way toward the end of this tail using pan-cancer approaches to identify ultra-rare driver mutations. Driver mutations in cancer, both common and rare, impinge on a core set of pathways. By coupling RNA expression profiling with DNA mutation sequencing, we can begin to interpret the implications of these driver mutations at the pathway level. This is extremely important in the context of a clinical setting where you may have a particular specimen that does not have a known driver mutation. Using the RNA expression readout to determine the state of dysregulated pathways can allow a clinician to make a determination as to the diagnostic state of the sample or which therapy a subject may respond to. One important class of specimens that can help to enable the discovery of pathway-level signatures are FFPE tumor biopsies. Over 20 million FFPE tissue biopsies are archived each year in the U.S. alone, and this class of samples is richly annotated with histopathology and response to therapeutics data. The RNA obtained from FFPE samples is heterogeneous in quality, and it is compromised along two principal axes. First, there are irreversible chemical modifications in the RNA, such as deamination and adduct formation. These modifications introduce sequencing errors, and they also reduce the efficiency of certain stages of RNAseek library prep. RNA from FFPE is also degraded and therefore cannot be poly-A enriched for RNAseek library preps. Alternative approaches must be used. The degradation is heterogeneous, as you can see in this plot on the right. I'm showing bioanalyzer traces for 3 different samples. You can see the extent of degradation varies across these samples. Both the compromised nature of FFPE RNA and the heterogeneity that we see in these cohorts introduces several challenges for RNAseek QC, library prep, and downstream informatics. Asuragen has developed an integrated whole transcriptome RNAseek workflow that is compatible with FFPE RNA. Starting from degraded RNA, we apply a QFI qPCR assay to determine the functionality of the material. After applying this assay, we subject the RNA to a ribosomal RNA depletion step, followed by RT and tagging. Strand-specific libraries are then amplified and then sequenced on an Illumina flow cell with a 2x50 paired-in reaction at a target read depth of 50 million reads per library. The resulting FASTQs are assessed by a bioinformatic QC process, and then a whole genome spliced alignment is performed. After performing this alignment, we break out into different types of analyses, 2 of which I'll discuss today in detail. First, we look at isoform and transcript quantitation from these alignments. We also perform mutation and indel calling, as well as fusion detection. Each of these analyses can be integrated and summarized under a compact reporting system that we have developed at Asuragen called Chirasika. The informatic challenges with FFPE RNAseek start with expression quantitation. Unlike intact RNA and fresh frozen, in which we can apply a poly-A prep, the RNA that we see in FFPE samples, we see an increased level of intronic sequences. And this is illustrated by this IGV plot at the top right of this slide. Here I'm showing for a particular gene the FFPE read alignments compared to an intact sample that was poly-A enriched. And you can see noticeably across the intronic regions of this gene, there's a pileup in the FFPE sample that's absent in the intact sample. And you can notice that this coverage is restricted to the gene body, suggestive of nascent or pre-mRNA and not genomic contamination. Most expression quantitation algorithms that have been developed to date do not take into account this high level of pre-mRNA. Asuragen has overcome this challenge by developing a robust expression quantitation and biomarker selection pipeline that identifies markers from this compromised material that generalize to other cohorts and migrate to other assays. A second challenge with FFPE RNAseek is the identification of driver fusions. Even outside of FFPE, the discovery of driver fusion events in cancer is a challenging problem due to the high disease-irrelevant background. This background is caused by several issues. First, we can have alignment error between two homologs that may look like a fusion but is actually a false positive. And we can also have a background driven by real biology, such as transplicing events that occur in wild-type cells, as well as passenger fusions that are not actually causal agents of the disease. These challenges are further compounded with the shorter fragment sizes that we see typically in FFPE RNAseek. To overcome these challenges, Asuragen has developed a very sensitive upfront fusion caller coupled with a cohort adaptive filter that eliminates most false positives while retaining high sensitivity. To date, we have applied our workflow over a wide range of FFPE qualities and mass inputs into the library prep. This is illustrated by this figure on the right, in which we performed a titration study using 3 different FFPE samples. All these samples were graded by a QFI assay, and on the top row, we have a high quality sample. In the middle row, we have a medium quality sample, and in the bottom row, we have a low quality sample. What this plot is showing is the technical concordance in the first column between 2 600 nanogram duplicates, and in the subsequent columns to the right, we're comparing one of those 2 600 nanogram duplicates to a lower mass input. We can see that across this plot, the coverage, normalized coverage between these 2 duplicates is maintained with a high R squared across this range of quality and mass inputs. You can see there is a tailing off of those R squares, but they all maintain a level of 0.98 or greater. As a more general statement, we've applied now our FFPE RNAseek workflow to over 500 clinical FFPEs with a success rate of approximately 90%. Next, I will discuss a case study that we applied this whole transcriptome RNAseek workflow to for the purposes of discovering novel diagnostic biomarkers in oncology. This case study focuses on improving molecular diagnostics for thyroid cancer. This is motivated by the fact that the incidence rate of thyroid cancer is growing faster than any other cancer. An early indication of thyroid cancer is a thyroid nodule, and these nodules are common in the U.S. population, but most are benign. The diagnosis of a nodule consists of an ultrasound-guided fine needle aspirate that is followed by cytology, and as cytology is indeterminate, molecular typing is performed, interrogating the presence of common driver mutations in thyroid cancer such as BRAF, RAS, taxate PPR-gamma, and RET-PTC. Many nodules are indeterminate by cytology and negative for these common driver mutations, which motivates the need for improved molecular diagnostics testing. In this case study, we applied whole transcriptome RNAseek to a cohort of 68 FFPE thyroid biopsies from surgical resections. Those biopsies were sectioned and assessed by a pathologist to determine the status of benign versus malignant, and further subtyped into different categories of follicular and papillary thyroid carcinomas, and for the benign samples subtyped into follicular adenomas, goiters, and nodular hyperplasias. All these samples, we also performed a Luminex assay to test for the presence of common driver mutations, including RAS, BRAF, taxate PPR-gamma, and RET-PTC. The pie chart on the right is showing the fraction of the malignant samples that were tested positive for one of these common driver mutations. Next, we isolated RNA from these FFPE blocks and performed whole transcriptome RNAseek. This is an assessment of the overall QC that we see for this cohort. The highest RIN that we obtained for all of these samples was 2.6, and this is typical of FFPE. We generally see most samples as being fairly degraded. Another QC metric that is being used more recently is the DV200 metric, and for this cohort we have 14% of the samples that show less than 30% DV200 metric. And for those of you who don't know, the DV200 metric is for a particular sample, and this is informed by a bioanalyzer trace, the fraction of RNA fragments in that sample that exceed 200 nucleotides in size. One of these samples failed the library prep outright, but all the other samples are shown below. The one sample that had a DV200 metric right at 30% is highlighted with the bold bar at the bottom. And what we're looking at in this plot at the bottom is where the reads are mapping to the genome post-sequencing. And you can see the sample with the DV200 right at 30% is mapping with about 30% exonic content, and it's sitting right in the middle of our cohort. All these samples that passed, that succeeded library prep, so excluding one sample, were moved on into the biomarker discovery phase of this project. We applied a variance filter to eliminate low variance potential biomarkers and arrived at a set of just over 21,000 candidate biomarkers to distinguish benign from malignant lesions. This is looking at now with a different view of that same PCA plot, the two principal sources of variation in the data that those candidate biomarkers are driving, and we see a separation here when overplotting the samples along those two principal sources of variation of the papillary thyroid carcinomas, which are circled in red in this plot. Those papillary thyroid carcinomas are separating from the benign lesions as well as the follicular carcinomas. And this is a very promising indication leading into supervised biomarker discovery and future selection. And this type of unsupervised analysis is something we typically apply just to get a sense of the data to see if the principal sources of variation in a candidate set of biomarkers is showing some type of class separation. Next, we moved into supervised biomarker selection and selected the top 25 biomarkers from this cohort that could distinguish benign from malignant lesions and could form a signature to classify benign from malignant lesions. I'm showing those 25 markers here in these two separate plots. The plot on the left is showing the FFPE cohort with each marker plotted in a different panel. The y-axis is the expression level of the marker, and the samples are broken out based on their respective subtypes. The benign samples are colored in blue and green, and the malignant samples are colored in orange, yellow, and red. You can see that some of these markers are showing upregulation in malignants relative to benigns, whereas others show downregulation. The plot on the right is showing the same markers from the perspective of the TCGA cohort, which is fresh frozen, and it lacks benign samples, but it does include, for some tumors, it includes a matched normal adjacent as well as a matched MET. So what this plot is showing, as you can see when you look at the same marker in both plots, is that markers that are downregulated in malignant samples are showing downregulation in the primary and MET relative to the normal adjacent in TCGA. And just to zoom in on one of those markers that's showing upregulation in a malignant set of samples in the FFPE cohort, here we're seeing a concordant upregulation in the primary and MET relative to the normal adjacent in the TCGA fresh frozen cohort. This trend and sign consistency of the effects was consistent for nearly every marker that we selected. And this is a very promising indication when leading into the next phases of potentially migrating this signature into a clinical assay to distinguish benign from malignant lesions in other cohorts. Now I want to take a step back and look at a different type of analysis that we apply to whole transcriptome RNAseek with the intent of identifying novel driver fusions. We applied our fusion calling pipeline to the whole transcriptome RNAseek data in this 68 sample cohort and identified a total of roughly 40,000 fusions. The caller that we use for the upfront fusion detection is highly sensitive, but it also includes many false positives, and it is highly sensitive to deal with the nature of FFPE, the shorter fragment sizes. Now, we eliminate most of these false positives through the application of a cohort adaptive filter that we apply, and this reduces that set of approximately 40,000 fusions by 3 orders of magnitude. To give you a sense of what the cohort adaptive filter is doing, we consider 3 different classes of fusions. First, we look at adjacent fusions, so fusions that are formed from 2 adjacent genes. These are often due to read-through events. We also look at intrachromosomal and intrachromosomal events. The plot on the right is showing the frequency at which different types of samples, benign or malignant, are covered by one or more of these classes of events. The fusions are ranked according to a score that takes into account the background frequency of the 3' and 5' partners in each fusion. And we set a score cutoff for each of these classes independently that enriches for the malignant, the presence of fusions in malignant samples relative to benigns using a Fisher's exact test to set the cutoff. This eliminates, as I said, the set of candidate fusions down to 41. All 41 of these were tested by endpoint PCR, and 26 of them were confirmed by Sanger sequencing. We followed up this confirmation by Sanger to test the presence of these 26 events in both benign and malignant samples using a more sensitive assay, and confirmed that 15 of these 26 fusions are in fact specific to malignancies. The remaining events are likely to be transsplicing events that occur in wild-type cells. Looking at the fusions that we confirmed from the perspective of the malignant samples that were previously negative for the common driver mutations, that 47%, we can see now that 2/3 of those previously mutation-negative samples that are malignant samples are now covered by one of these fusion events that we detected. Of that set, 17% of those fusions have been published either in thyroid cancer or another cancer type, but 50% of them were novel. And those novel fusions, the 3' genes in those fusions, are very well-known oncogenes and known driver fusions, the driver mutation genes in both thyroid cancer and other cancers, suggestive that many of those novel fusions are in fact driver fusions. Of the one sample type, or rather fusion type that we knew to be present in these samples, which is Pax8 PPR gamma, this was present in 3 of the 68 samples. We were able to detect this fusion with 100% sensitivity and specificity using the pipeline that we've applied, which is the sensitive fusion caller followed by the cohort adaptive filter. Moving on to recap what we have learned in this section. First of all, given the right combination of molecular and informatic processes, high-quality data can be obtained from FFPE tumor biopsies. And we've demonstrated that through the application of this case study in thyroid cancer, where we apply whole transcriptome RNAseek coupled with Asuragen's biomarker discovery pipeline to identify expression biomarkers in FFPE specimens that generalize to other cohorts, including fresh frozen biopsies. Finally, we can apply Asuragen's fusion pipeline discovery process to whole transcriptome RNAseek to enable both the reliable detection of known fusion events as well as discover novel driver fusions. Now, moving on to part 3. This is part 3, connecting the dots. In this segment, I'll discuss how to navigate the transition from preclinical exploratory discovery work to a clinical assay using next-generation sequencing. The traditional path has been to perform discovery on a high-content platform, such as a microarray, and then migrate a set of biomarkers or signature onto a smaller footprint assay, such as qPCR. The clinical assay often uses a different isolation protocol, sample prep, and quantitation chemistries, and this results in platform-specific biopsies and technical variances, differences in the dynamic range that ultimately can lead to the loss of markers and degraded signature performance. Next-generation sequencing offers the opportunity to de-risk this process by unifying the discovery and clinical assay development stages. Asuragen enables this unification through Chirasika targeted RNAseek, which shares several common steps with whole transcriptome RNAseek. First, it uses a common isolation protocol to isolate from either FFPE samples or a fine needle aspirate. It also uses a common RT reaction to convert the RNA to cDNA. And then it also shares the actual measurement technology that these libraries are destined for, which is a next-gen sequencing flow cell or a PGM. As a case study to illustrate this migration path from whole transcriptome discovery and using RNAseek and FFPE samples, and the migration of that signature that was derived from that study to Chirasika targeted RNAseek assay. Here we have a cohort where we selected the 25 diagnostic biomarkers to distinguish benign from malignant lesions in thyroid cancer. Those 25 markers, along with 15 additional genes, were migrated to a targeted RNAseek assay using Chirasika. The additional 15 genes included endogenous controls and markers of theranostic value. The Chirasika targeted RNAseek workflow is as follows on the right. We start with isolated RNA and perform a qPCR-based QFI assay to assess the functionality of the material. This assay is performed post-RT. That RT is then fed into a targeted multiplex PCR and tagging step. Those libraries are then amplified and then sequenced on a next-gen sequencing instrument. The 40-gene panel that we developed using the Chirasika targeted RNAseek custom assay design was applied to two different cohorts. First, we applied it to a cohort of 123 FFPE samples with known pathologies. This included the original 68 samples that we used to discover the diagnostic signature, and the prevalence of this cohort was 69% malignant. The second cohort that we applied this targeted RNAseek panel to was a set of 65 fine needle aspirates for which surgical outcome was known. The overall prevalence of this cohort was 56% malignant. We applied, after performing the targeted RNAseek assay to both of these cohorts, two different types of analyses. First, we looked at the comparison of whole transcriptome RNAseek with matched samples for targeted RNAseek to ensure that there was good analytical concordance between the discovery and the clinical assay. Secondly, we looked at the diagnostic classifiers that we could build from the targeted RNAseek data, looking at classifiers built on FFPE samples and then tested on FNAs. This plot is showing each of the 25 diagnostic markers that were selected from the whole transcriptome FFPE cohort, with a comparison of whole transcriptome RNAseek along the y-axis compared to targeted RNAseek along the x-axis. Each of these is a different marker, and some of these markers actually are more functioning as endogenous controls. You can see there's one marker that has lower variance in the center of this plot by both technologies. But what we can see is that nearly every single one of these markers is showing very good concordance between the two platforms, the discovery platform and the clinical platform. Next, we looked at both cohorts by projecting the markers that we measured onto a PCA biplot, where we're separating both the markers by the principal sources of variation in the data, as well as the samples, and those are overplotted in the colored squares. One thing that stands out in this plot first is that we see a separation between the benign samples and the malignant samples, and this separation is achieved for both the FNAs as well as the FFPEs. And the point at which those samples separate, which is right at the dotted line, is roughly the same for both cohorts and collection methods. The second feature that we see in this plot is the separation of the FFPE samples shown on top and the FNAs shown in the bottom. And this separation, upon closer inspection of the markers that are involved in PC2, we found those markers to be present at high levels in blood, and this is consistent with the fact that FNAs are more prone to blood contamination than FFPEs, which is what drives this effect. Finally, moving into the construction of several different classifiers using this data. The first classifier that we developed, which is shown in blue, was trained on the FFPE cohort, the 123 FFPEs, and then tested under leave one out cross-validation on that same cohort. The accuracy that we're able to achieve with this classifier is quite good, with an AUC of 0.94 and impressive sensitivity and specificity numbers as well. The corresponding ROC curve is shown highlighted in blue in the bottom left. And this finding is not actually that surprising, given the fact that about half of this cohort was used to perform the initial diagnostic biomarker selection. So there is a bit of bias baked in there. The second classifier that we built was trained on the FNAs and then tested on the FNAs under leave one out cross-validation. And this result is actually telling us something quite interesting. First of all, it tells us that given that the high AUC sensitivity and specificity numbers are retained in this cohort, it tells us that the discovery process of these markers was successful. We identified markers that have diagnostic utility in FNAs, and those markers were discovered in FFPEs. And then finally, looking at the last classifier that we developed, which is highlighted in green, this classifier was trained on the FFPE cohort and then tested on the FNAs. And this result, I think, is the most compelling, given that this classifier, both in terms of the content, the markers that are included, as well as the coefficients of the model, have never seen, this model has never seen a fine needle aspirate. It's only been trained on FFPEs, and FFPEs are what guided the selection of those markers. And in this classifier, we were able to retain very high AUC sensitivity and specificity in the fine needle aspirate cohort. So moving on to wrap up this section with what we have learned. First of all, Chirasika targeted RNAseek is both quantitative and analytically concordant with our discovery technology, whole transcriptome RNAseek. Secondly, signatures that are discovered, developed, and trained on FFPE biopsies using whole transcriptome RNAseek and then migrated to target RNAseek show clinical value in FNA biopsies. Finally, Chirasika is what enables the unification of the discovery and development phases of diagnostic pipelines. And with that, I'll hand it back to Gary for part 4.
Part 4, putting it all together. So in this segment, we're going to talk about how we've been able to take DNA markers and RNA markers, including novel mRNA expression markers and translocation, and marry them with a unified total nucleic acid-based clinical workflow. And I'd like to focus today on a clinical case study around thyroid cancer, but I think it's important to note that our general strategy and approach here is relevant to many different cancers and pathologies. Let me begin with an overview of this particular unified NGS workflow. And I'd like to point out a couple of key elements as we go along. Since this is a thyroid clinical case study, it's important to provide some context on the acquisition of the sample type, which in this case is a biopsy. It's taken as an FNA from the patient. We're able to utilize our RNA retain technology to preserve the sample, which is critical because it includes RNA and DNA markers. And then we can use the QFI assay to partition quantification of both the DNA and the RNA load within the total nucleic acid sample. And that really provides the first innovation here. We have the capability to take a total nucleic acid, which relieves us from the requirement to independently isolate DNA and RNA, and instead individually query how much DNA and RNA there is in a DNA sample and use it accordingly. We can then pass that material into the enrichment portion of the workflow, and at that stage, since we're looking at multiple different types of markers and we're building a larger panel, that we have the opportunity to ask multiple clinical questions. So that's another innovative feature of this particular case study, and that is that it taps into multi-purpose clinical content. Not just diagnosis of malignancy, but also markers that can have informative insight into uptake of radioactive iodine for patients with thyroid cancer, for potential prognostic implications for these patients, for targeted therapy and theranostics, etc. Once that's been sequenced on the appropriate NGS instrument, it goes into the integrated bioinformatic pipeline. So going through our variant caller pipeline and the corresponding Chirasika package for analysis, then that's really the third key innovation here, because we're looking across different pathways. We're integrating a constellation of different types of markers, from DNA mutations and indels to CNVs to fusions and translocations and mRNA expression markers, all of which can be synthesized into an evidence-based report that can be passed on to be actionable at the level of the patient. So in terms of the design of the panels themselves, since we're interrogating both DNA and RNA, we have opportunities here to be able to build the content in a way that provides some additional benefits. For example, in the DNA pools, we're targeting a total of 20 genes. So those genes are represented in loci spread across 2 independent pools that are utilized for enrichment. However, an interesting feature of this that we've built into the assay is the ability to have overlapping content between the 2 DNA pools. So some of the most critically important genes that are relevant in the clinical context of thyroid cancer, like BRAF and the RAS genes, are so important that it's vital to be able to make very accurate calls on these particular genes. So we've designed the targets to be completely independent in primer designs across the 2 pools so that we have built-in confirmations associated with the outcome of the DNA variant calling. On the RNA side, we have also 2 pools. One of these is dedicated to query translocations. There are a total of 52 translocations. And one of the RNA pools and the other queries 40 mRNA markers for expression. And in a similar vein, we have controls that overlap between the 2 pools to ensure integrity in the results and interpretation. So we're covering a broad range of different content for DNA and RNA in a workflow that ultimately puts these 2 together that create a very straightforward approach for routine use. Let me highlight then a few other novel features of the assay and how we can now really put it all together with these different innovations that spread across the pre-analytical, analytical, and post-analytical components of the workflow. So let's consider something like total nucleic acid that we're going to analyze from the sample that's taken from the patient. In this case, because thyroid biopsies often don't yield a great deal of total nucleic acid, it's important that you don't overtly consume that material in the QC assay. And as a result, it led us to develop our QFI DNA assay to be even more sensitive. So what's shown in this plot is the advance from our version 1 of the assay, which is shown in blue, to version 2 of the assay, which is shown in red. What we've done is we've changed our approach here that allows us to have much higher sensitivity and achieve accurate quantification to less than 0.05 genome equivalents. So now we can use an absolute paucity of material in the QC portion and preserve as much material as necessary for the downstream analysis. Another way we've used this assay is that it allows us to do a very strict molecular accounting of both DNA and RNA in a way that can inform the bioinformatics and help us understand what our call thresholds can be. Take this example. This is a 5% BRAF V600E SNA that has been embedded in the background of wild-type DNA. And what's varying is the total copy number of DNA across a series. So the background of 5% variant is held constant. So what that means is that 1,000 total copies at 5% variant, we have 50 mutant copies of BRAF, and so on down to 63 total copies and only 3 copies of BRAF mutant. We know this because we've been able to analyze it quantitatively using QFI DNA assay. So now if we look at how this material calls in the downstream NGS assay, you can see that we're able to maintain a strict quantification of this 5% level at the higher inputs all the way down until we get to about the 3 mutant point. So you can see these are done in replicates, and we waver with a dropout at the lowest input, which is only 63 total copies, or 3 mutant copies of BRAF. So at that point, we're really getting to a stochastic range. But we have the ability now to use this information to help inform exactly what our stability is for variant calling, even for low quantity samples. What about on the RNA side? We've used a number of different statistical methods to be able to identify what's an appropriate RNA normalizer for thyroid cancer. We've identified that marker and put it to good work in a QFI RNA assay. The plot shown at the bottom here is a representation of different inputs of total nucleic acid and the corresponding copy number of that vetted RNA target that's very stably expressed across a range of different thyroid subtypes of cancer and benign conditions. If we then take that information and look specifically at particular fusions that may be detected that are clinically relevant, in this case a PAX8 PPR gamma fusion in one clinical FNA, and in the other case a RET-PTC fusion, we're able to show that down to about 1 to 5 nanograms of total nucleic acid allows us to achieve a ready detection of these particular events with a corresponding copy number of our target, which we can then standardize as a consequence of input into the assay. In analytical performance, going through the enrichment process and really vetting the assay on a larger scale, we can look at metrics associated with sensitivity and PPV. This plot shows you the corresponding analytics for DNA detection. It's not just cancer cell line controls. In this case, we're looking at 72 different specimens, of which 69 are either benign or malignant clinical FNAs with known outcomes for mutations and other variants. And based on the precision recall curve, you can see we generally have about 90% sensitivity at 90% PPV. So it's a very high performing assay down to about 2% variant detection. One of the interesting features of this approach, as I alluded to, is the fact that confirmation is sort of built into the assay. We're running 2 assays in one for many of the most clinically relevant targets. And a couple of examples of this. On the DNA side, this data that's shown on the current slide is a KRAS Q61R mutation that was detected at a very low level, about 3.5% within one of the clinical FNAs that we tested. And in the corresponding DNA pool to this one with the orthogonal design, it was detected at 2.99%. So high read coverage and a great deal of confidence. As you can see, these stand out very clearly in the pileup and IGV. We've also done this on the RNA side. So translocations are a critical part of being able to correctly call these particular specimens and index them in a clinical sense. And one of the more important translocations that's associated with thyroid cancer is PAX8 PPR gamma. So since we have a translocation pool, we have an mRNA pool, we can use both of those pool designs to our advantage. We can first detect the translocation, for example, as a PAX8 PPR gamma fusion, but then we can see confirmation by virtue of its 3' partner of that fusion, the PPR gamma mRNA. In the mRNA enrichment pool, we can see that it's specifically elevated in cases, and only in cases, where the translocation is present. So the plot here shows in red those that have the translocation and those in black that do not, and the points that really stand out in red are those that have very high expression of the fusion partner as expected. So now in the post-analytical part with the bioinformatics that can interpret across many of these different types of markers and put this into a broader perspective, I think I'd like to just wrap up with a clinical study that we did that highlighted the value of these many different types of markers. So in this particular clinical research study, we looked at 50 indeterminates of fine needle aspiration samples by cytology. So these are samples that had not given any type of clear indication of cancer. And many of these patients, in fact, end up going on to surgery. We tested them with a 17-marker probe-based assay that represents very well-established markers by the American Thyroid Association, conventional markers in DNA and RNA. What we observed from this cohort of 50 different indeterminates is that we could call about 75% of the malignant samples. And these were split out by BRAF status, RAS status, or with translocations like RET-PTC or Paxate PPR gamma. However, there's a black hole here of about 26% that is unexplained in malignants. So all of these samples are in fact malignant, and yet the conventional markers only cover about 3/4 of them. How does that compare with our 104 gene comprehensive panel that spans many more DNA and importantly RNA markers, and most importantly spans many different categories of markers? In this case, you can see we can seal the gap and we can explain the dark matter of this hole that's unexplained with conventional marker set. So we're still calling these, of course, the BRAF and the RAS and the conventional translocations, but now we can close this gap of malignancy with novel translocations, novel mutations, and importantly, novel expression markers, so that we can explain almost the totality of this pie. In addition to having a very high sensitivity, the assay does have a high specificity as well, and where we did see lapses in specificity, about half of those that were called were associated with RAS or Paxate PPR gamma translocations, which have been observed by many others in other samples that may be pre-malignant. So this technology for thyroid cancer has been licensed to Interpace Diagnostics, and it represents the real power of integrating across many different marker types for a specific cancer, and is applicable, I think, to many other cancers and pathologies. So I'll wrap up on this segment by commenting on the value of this particular approach to maximize workflow efficiencies. This isn't a bolt-on of an RNA panel with a DNA panel. This is a truly integrated system that was built from the ground up to accommodate multiple types of markers across both of those nucleic acid types. That it can synergize interpretations and provide intra-assay confirmation, which is a powerful feature for having confidence in clinical outputs. That you can get diagnostic value by looking at pathways at a much broader level than just individual mutations, and that ultimately this approach can advance drug development and clinical trials by taking the most informative content from DNA, from RNA, etc., and putting it into a validated assay configuration that can be amenable in a workflow for routine testing. So at this point, we'll conclude and we'll wrap up with some Q&A on the webinar.
So moving into the Q&A session, looks like the first question relates to RNAseek, and it is, how does your FFPE RNAseek workflow compare to the Illumina Access FFPE RNAseek assay? So to answer this, first off, I should say that we have not done a head-to-head comparison between our workflow and the Illumina Access kit. As I understand it, the Illumina Access kit works by selective enrichment of exome sequences through a capture-based approach. However, I do know that the metric they used for pre-sequencing QC is based on the bioanalyzer percent DV200. And from our experiences with our workflow, what we find is that the percent DV200 is actually not very predictive of the post-sequencing data quality, at least in the context of our assay. And this may be related in some way to the capture that's used, but what we find is that many samples that fall below the cutoff that's recommended for access, which is the 30% DV200, we get many libraries that come in at reasonable and high quality in spite of their low DV200 score. So I can speak to that aspect of it, but I think a head-to-head comparison between several different approaches for FFPE RNAseek would be warranted in the future to understand which is the most appropriate approach for different clinical applications.
Okay, one question that's come in is that other than thyroid cancer, what disease areas do you think that a combination of targeted DNA and RNA sequencing would provide value? And I think this is a good question because the type of approach that we've described is applicable to a number of different types of cancers. A good example would be lung cancer. In this case, we know that there are a number of mutations that have been well-defined for EGFR, for KRAS, and other targets that are highly actionable for targeted therapies, but we also know that there's an emerging landscape for translocations with the ML4 ALK, as well as ROS and RET fusions, and that these are also highly actionable. So looking more comprehensively at how these could be put together, not just disconnected workflows and approaches, but an approach that looks at lung cancer specifically and builds a clinically minded assay that can put together both types of these markers in a way that's representative of how a personalized medicine of lung cancer is being addressed. It's something that could be very powerful for the future. Another target that's of relevance to several different pharmas is P53. And so you think about P53 in terms of an exon sequencing assay, but in reality, I think there's a lot of power in pulling together both DNA and RNA in that context as well. Because we're looking at not only the potential for a mutation in P53 that can lead to an inactivation of the protein and a consequence for those individuals and patients, but also the fact that you have opportunity to look at a number of different isoforms of the expression of P53, as well as other markers and expression markers in particular that may be in the P53 pathway and affected by differences in P53 relative activity. So this can also help maybe unravel some of the complexities of how P53 manifests in different cancers and with its tissue selective expression and things of that sort. Well, we allocated an hour for this webinar, and we're running up against time at this point. So I want to thank everybody for their participation. We received a great many questions, and we will certainly be following up with each and every one of those. On behalf of Asuragen and LabRoots, thank you again, and have a great day.